I'd like to say good morning and good day and good evening. It's really my pleasure to welcome you to the third International Cohort Summit of the International 100K Plus Cohorts Consortium. Next slide, please. My name is Jeff Ginsberg. Advance again, please. And I'm at Duke University, and I'm co-chair and founder of the Global Genomic Medicine Collaborative and of the IHCC, the International 100K Plus Cohorts Consortium. I'm joined by my two IHCC co-chairs, Barry Manolio from the National Human Genome Research Institute at the NIH, and Peter Goodhand of the Global Alliance for Genomics and Health. We expect to have approximately 150 people from more than 25 countries participating in this conference over the next 2 days. Next slide, please. As you know, this meeting was meant to be held in Santiago, Chile. That choice was not only because it's a cosmopolitan city, but also to recognize the potential for the IHCC to engage with cohorts in regions of the world with limited resources. And for so many reasons, I'm sure we all wish we were there. And I want to thank, in particular, the members of the planning committee who were engaged in having a meeting in Santiago, and in particular, Ricardo Verdugo from the Universidad de Chile for his help in planning this face-to-face meeting and also for pivoting to this virtual one. Thanks for my colleagues on the planning committee and also in particular the local individuals from South America. You may recall this IHCC vision was created right after the first summit almost 2 years ago to the day, and the vision is to create a global network of large cohorts with multidimensional data from diverse populations for translational research that will be maximally utilized to enhance scientific understanding of the biological, environmental, and genetic basis of disease and to improve population health. Next slide, please. You will also recall that the IHCC was started on the premise of combining data from these large cohorts would enable us to address pressing global issues and questions that no one cohort alone could answer. That these activities would enhance the values of the cohorts and leverage the enormous amount of investment that had already been made for them. Our criteria for membership has been cohorts having greater than 100,000 participants, unselected for disease, having available biospecimens now or in the future, and the potential for longitudinal follow-up. And as you will see in our charter discussion later in the day, we have kept to these criteria, but have also made some exceptions that allow us to optimize the diversity in the populations that could be represented by the IHCC. Next slide, please. Since late 2018, we've had three main work streams. The first was to develop an information technology platform that would allow for cohorts to be effectively discovered by the scientific community and to promote sharing of data. And as you'll see later on in this meeting, we will report on the development of an atlas that is the first version of this opportunity to discover cohorts, as well as the architecture in a federated form that will allow for cross-cohort collaboration. The second work stream has been one around developing a robust scientific agenda that leverages cohort collaboration and maximizes input from the global scientific community. And you'll also see reported out at this meeting later on the first demonstration of multi-cohort scientific activities that are meant to really just to demonstrate the absolute power of cross-cohort science. The third work stream has been around policy, and policies in particular that allow us to seamlessly, if possible, share data, to engage with industry partners, and also to begin to put together our strategy for publications and the policies around that. And all of these are meant to really lower the barriers to global scientific collaboration and also translational science. I should mention at this point that we've had a very engaged steering committee representing the leadership of these work streams, and you'll be hearing from all of them later in later sessions as they are moderating a good part of this meeting. The next slide, please. We've also been very busy, the steering committee in particular, in other activities over the past year that will be covered in greater detail a bit later today. We've developed a charter that provides for structuring the IACC, its membership and decision-making. We have also recently turned our attention to COVID-19, as many other research organizations have done, and will be engaging with you to develop a strategy that absolutely leverages the power of the cohorts in the IACC. I'm pleased to report we've also secured funding from the National Institutes and from the Wellcome Trust to support many of the activities we will discuss at this meeting, and also we will continue to support additional funding activities as the year goes on. And this week, we are launching a process to develop a 5-year plan that we hope will be completed by the end of 2020. Next slide, please. I want to acknowledge that we continue to grow and welcome new cohorts that have joined the consortium. I'm also pleased to say that the IACC participated in a meeting last month to plan a Pan-African cohort, provisionally called the African Population Cohort Consortium, that is aimed at accelerating health and well-being throughout Africa, and that will also be led by African scientific leadership towards discovery and impact. The APCC effort has been spearheaded by the Wellcome Trust along with several other national and international funders, inclusive of the NIH, and you'll be hearing more about the APCC in coming months. Next slide, please. The IACC now has representation across the globe, but there are still several gaps in South America and also on the African continent. We now have 97 cohorts, and you can find these on our website. These represent greater than 50 million participants worldwide. Next slide, please. So our objectives for the next 2 days in the International Cohort Summit number 3 is to galvanize this organization around a visionary charter and path forward, to examine how the IACC can rapidly mobilize worldwide cohorts to address the COVID-19 pandemic, to introduce the IACC to a cohorts atlas that can be used to stimulate and enable collaborative activities across the cohorts, and to engage the entirety of the IACC membership in developing key topics that will chart a scientific agenda that can only be achieved by assembling cohorts and their data. I am quite optimistic that we'll be able to achieve these goals during our virtual summit. And lastly, I wish to thank the amazing team that has made all this possible. The IACC is extremely lucky to have this diverse group of talent to support it. And I think I can hear you clapping virtually. I hope you can. I hope you are. And so at this point, it's my honor to introduce our first keynote speaker, my colleague, Dr. Francis Collins. His bio and those of all the speakers are in the meeting booklet, so I will not repeat it here, but I do want to first say 2 things. First, thank you, Francis, for your vision, for instigating the IACC and your continued and unending support of what we're doing. And also, the second thing is that we learned on Friday that you were announced as a foreign member of the Royal Society, a tremendous honor, and we congratulate you on that, and we'll certainly add that to your bio. And now, welcome Dr. Francis Collins, the Director of the National Institutes of Health.
Well, thank you very much, Jeff, and thanks to you and Terry for your leadership of the IHCC. And it's wonderful that we're at the 3rd annual meeting, even though not quite in the format that we had all hoped for in Santiago. I think I see on my banner at the bottom here, there are 89 participants currently taking part in this virtual meeting. Wonderful that people have figured out ways to do that from all over the world. I think I saw in the chat box, hello from Germany and hello from Japan. Welcome, everybody. I'm here in my home office in Chevy Chase, Maryland, where I've pretty much been continually for the last 6 weeks, other than one brief excursion to the White House, and I don't want to talk about that. Otherwise, my work here has been very much in the space of trying to drive what we can do at NIH about the COVID-19 crisis, which of course is occupying the minds of virtually everybody working in medical research around the world. Most of our staff at NIH are working from home. I see Terry in a location there that looks like it's probably her living room. That is where everybody has been hunkered down unless they were involved in direct patient care. And we do have a hospital, the clinical center, which continues to take care of patients who are in urgent situations. And of course, we also have the Vaccine Research Center at NIH, which has been very much on the forefront of trying to prepare the vaccine, which we hope will be ultimately the way in which we deal with this crisis. There are now at least 5 vaccine programs that are under development and moving at pretty good pace, which is good to see. So I'm sorry that we can't have the opportunity to gather together and face-to-face to talk about the IHCC and all the progress that has been made, but I'm glad that it was possible to put this together as a virtual meeting and have the kind of very robust agenda assembled that your leadership has managed to put together. I just got through my workout, so if I'm sweating a little bit, it's not because I'm nervous about talking to all of you. It's still just after 7 in the morning on the East Coast of the U.S. So I thought I'd say a little bit about what's occupying the attention of myself and my colleagues about COVID-19, but then I want to talk about IHCCs and its potential role in this space, which I think could be quite significant. Here at NIH, we are engaged, as I just mentioned, in an effort to try to accelerate the development and deployment of vaccines. The vaccine that was developed in our own Vaccine Research Center and now done as a collaboration with a company called Moderna got into phase 1 trials 63 days after we first saw the sequence of the RNA virus called SARS-CoV-2, and is now about to move forward in what we hope will be a combination phase 2-3 trial as early as July, with the intention of trying to get this to the point where we could say whether it works by the fall. That would be a lot faster than has previously been imagined. And of course, then you have the manufacturing scale up to deal with. And we are very sensitive to the fact that this is a vaccine, if successful, needs to be available to the whole world, not just to the US. There are 4 other companies that are pushing forward vaccines that are also looking promising. And so it's even possible we might have the opportunity for a master protocol that does a comparison of one vaccine to the other side by side, which would be a really good thing to be able to do. Lots of running around to try to make that happen. In addition, we're working intensively on trying to identify the next therapeutic opportunities. It was good to see that we have at least one drug now that in a randomized placebo-controlled trial was able to shorten hospitalization and had a trend towards improved survival. That's the drug called remdesivir. But of course, that's not a home run. It's a beneficial drug, but we need to have things to make it even better. Maybe we'll need combination therapy for this virus, just like we do for viruses like HIV. So we have organized something called a partnership called ACTIVE, which stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines. And that group, which has been meeting since April the 3rd, is this week trying to come out with a prioritized list of what the next therapeutic compounds would be that would be most important to get into clinical trials as soon as possible. Trying to take amongst 170 possibilities, what are the ones that maybe ought to be at the top of the list? We'll see what happens later this week when they come forward with that prioritized list. And we're also, of course, trying to organize all the clinical trial networks to be sure that they're ready to test these particular therapeutic ideas when they come forward. Some of those will be antiviral, small molecules, and some of them will be immunological therapies like monoclonal antibodies, and some will be therapeutics aimed at the people with the most severe lung disease, where the suspicion is that the immune system is partially contributing to the severity of the disease, and perhaps such things that deal with cytokine storm might be actually beneficial. All that's going on in this partnership, which involves 17 companies, the NIH, the FDA, the CDC, the European agency that's equivalent of FDA, as well as BARDA, as well as a lot of academic institutions, all managed by the Foundation for NIH. Yeah, this has been all-consuming as well. And then on top of that, we have been given a mandate by the Congress to do something to speed up the possibility of diagnostic testing so that it's more available and in point-of-care fashion, as opposed to the kind of testing that has to be sent off to a central laboratory and then you wait for the result to come back. Because we need to know, as people begin to come out of seclusion and back into public spaces, who has got the latest version of this infection and what should we do to quickly get them quarantined and chase down their contacts. So we are working on that in a rather unanticipated way and a rather unprecedented way by inviting all those technology developers who have great ideas about how to do diagnostics in point of care to come forward in what we're calling a Shark Tank, have their ideas tested out, and if they look promising, give them the resources and the advice in order to speed up the process of commercialization and deployment. So that's on the plate as well. All of that is all rather consuming, but it's the right thing to do. And I know everybody listening to this in your own way are also pulling together to try to address this global pandemic. How can IHCC help? Well, in some very important ways. When you look at the consequences of what happens to somebody who is exposed to this SARS-CoV-2, it's clear there's a lot of variability between the consequences. Some people who get infected but never even know it, completely asymptomatic but capable of passing the virus on to others. People who get very mild symptoms, and then people who get extremely ill. People who end up in ICUs on ventilators, and sadly, many of them also lose their lives to this disease. And we know that some of the risk factors are chronic illnesses and older ages, but that's not sufficient to explain the high degree of variability. IHCC is in a great position to be able to contribute to that because of your reach across the world. And certainly the focus that has been baked into this, which is to look at both environmental and genetic factors, means that IHCC has an opportunity to really identify with the appropriate power and numbers what are the factors that play out in terms of risks of severe disease. And alternatively, what are the factors that pay out in terms of resilience? Why do some older people with chronic disease do just fine? What is that protection about? If we knew that right now, I guarantee you it would have all kinds of consequences for the way in which we're trying to prioritize drug therapies, because it's been proven over and over again, if you have a natural variation that provides some indication about a disease risk, that's a very good place to go if you're trying to develop a drug therapy. I wish we had that answer right now, but I know many people are starting to work on it. And more than that, I would say not just about susceptibility, but what about variations that play out in terms of drug responsiveness? I mentioned remdesivir and the fact that it does provide some benefit, but not equally to everybody. So what's that about? Is there some pharmacogenomics there that we ought to be looking at in terms of who responds well and who doesn't? And even with vaccines, which are coming along, as I said, in sometime in the next 9 months or so, are we going to be able to identify those individuals for whom the vaccine is particularly effective or particularly ineffective? Because I would be surprised, no matter how optimistic you are, if this vaccine is 100% effective for everybody. And we need to understand what those modifiers are in order to be able to adjust accordingly. So all of this seems to be like a great place for the IHCC to be positioned. And I know you're going to be talking about that in the course of this meeting with special session on COVID-19. Indeed. We're even contemplating right now whether some of these cohorts might also be in a good position to be volunteers for testing out the vaccine, since they're often people who have been, at least in some of the cohorts, consented for recontact. And if we want to find out whether a vaccine works in various parts of the world, maybe this is not a bad place to think about starting. I hope you'll talk about that as well. So I wish I could listen in to the entire discussion that you're going to be having. It's going to be a bit of a busy week here. I have a Senate hearing on Thursday and a few other things that are going on that are going to be quite consuming. But I know the way the agenda has been planned is going to bring forward a lot of important ideas and a lot of consequences I hope will follow, because I really think IHCC at this point has arrived at the juncture where it's about action, about actually moving things forward, figuring out how to share data, dealing with the information challenges and dealing with the policy issues, and going forward with some specific projects to show how this can work. I'm delighted to see there's a focus on getting a broader distribution of cohorts around the world in places like South America and Africa. Hope everything will be done to try to push that agenda forward. And I want to thank the Wellcome Trust, who've been such a wonderful partner with NIH in the financial support of this effort and providing what I hope you will see as a bit of a stable base for this IHCC effort. Again, NIH is very interested in continuing to see ways to support that, and we hope you will look forward to any kinds of announcements we have that might be appropriate for IHCC to consider. We would, of course, love to see more competitive grant applications coming through at the national and international level to support this effort. So you all have unique qualifications, capabilities. You can be out in front there as pathfinders for where we really want to go in terms of understanding genetics, environment, health, and illness. And it is a wonderful thing to be able to see, now at this third meeting, how much of the world has come together to try to make those kinds of dreams come true. And I really appreciate your determination to stick to that, even in the face of what is otherwise a pretty daunting circumstance of COVID-19. So I hope you have a wonderful summit. I'll look forward to hearing all about it from leadership as you come to a close and put forward all the next ideas that you want to carry forward. And I just want to tell you personally, from me, I'm one of your biggest supporters and want to see this effort be as successful as it possibly can be. So with that, I'll stop and see if there are questions that people want to pose. I see that there is a chat box here, and so people could use the chat box to ask questions unless you have some other plan, Jeff and Terry, about how to do this part of the meeting.
Thank you, Francis. We definitely want to respect your time, but hopefully you'll be able to stick around for a few minutes for some questions and answers. And there's some instructions now that have appeared for our audience to raise their hand and ask you questions. We'll make sure the team will identify who is raising their hand to speak, but maybe while people are thinking about that and understanding the process, I'll just ask the question about that you alluded to this. In April you put together this group towards developing novel therapeutics. The ACTIV group, I think, is what you called it.
Yes.
I'm just wondering, since you're so connected to other leaders of research organizations like the NIH across the globe. What have you discussed, if anything, around bringing them together to really harness, what this is obviously a global issue, and we don't want to see, a lot of duplicative research going on worldwide. Have you had any conversations that indicate to us how the research community across the globe is coming together?
Yeah, great question. And we have indeed, particularly with the UK, where there's a similar effort underway to try to harmonize and prioritize the therapeutic options. So in conversations with Jeremy Farrar, well known to all of you, and Patrick Vallance in the UK, we've agreed to share completely openly what they're doing with something called Accord and what we're doing with ACTIV. We also have good connections with Europe. And again, because ACTIV involves these 17 pharmaceutical companies, most of which are international, that also links us up with things that are happening outside the US. We have a good connection also with the IMI through Paul Stoffels and J&J. So yeah, we are trying very hard to connect all these efforts and not be duplicative. It's a great point.
Thank you. So again, raise your hand. It looks like that we have somebody raising their hand, and I'll let our team identify who. It looks like Terry Manolio.
Thank you. What do you know? So I wonder if you could say a little bit more about your comment on the cohorts potentially being a source of participants for clinical trials, either of the vaccines or of drugs. So how would they go about, addressing that or signing on to participate?
And it's a very nascent idea, and it will depend, of course, on exactly how those cohorts were set up in terms of what the recontact potential was. I have been thinking about this with regard to the All of Us program in the U.S., because there we have a very diverse population geographically and in terms of ethnicity and race and also socioeconomic status, exactly the kind of group that you'd really like to be able to get involved in testing out a vaccine, and people who have been consented for recontact and who are being cared for, many of them through health provider organizations or community health centers that could be involved in getting engaged in administering a test vaccine. It's an idea. We're just beginning to explore this to see if that might be a way, because these trials for vaccines, you're going to want to enroll tens of thousands of people, because you need, in order to see whether the vaccine has worked or not, you need to do it in a place where the virus is still circulating, and you need to have enough power to be able to tell after a month or two, whether the vaccinated people had fewer cases. And you quickly go through the math, and you need a pretty big study. So rather than starting totally from scratch or trying to work with some of the existing clinical trial networks, which are also in many instances good sized, if we have these cohorts and if they've been consented and if they're motivated people who are our participants, maybe we ought to engage with them. I'm just throwing this out there as a somewhat half-baked idea, but it is worth considering, I think, for people who are trying to set these things up.
Thank you. I don't see any other hands raised, but I maybe I'll also just, since you happen to be a genomics person, Francis, I wonder if you might comment on the opportunities to harness and thinking also about the IACC role, which we'll talk about later in the session, to harness the power of all the sequences that are available worldwide and linked to the clinical data for COVID-19 patients. I think that's a great opportunity. Perhaps you do, and think about that as also a way that we might scale the IACC's activities to, obviously to other diseases, using COVID-19 as a demonstration project, if you will.
So with COVID-19, certainly if people already have sequence data and information about the clinical experience, one would want to know right away, is it enough there that one could start to do a GWAS to understand if there are variants that play a role in the likelihood of severe infection? I guess we all have our ideas about favorite candidate genes that might be involved there, like ACE2, since that is the receptor for the virus, or TMPRSS2, which is a cofactor. But, our guesses are usually wrong when it comes to these things, which is why having enough power to do a genome-wide association would be really useful. And I guarantee you, if we had that data right now and we could crunch it, we would probably learn something about the viral pathway for causing this disease that we'd want to know and that might have immediate consequences for therapeutics. Because we don't just want to know about the viral genome, although we want to know that too. We also want to know about the host genome. Just speaking of the viral genome, that's another thing where we have lots of sequencing capacity involved in IHCC. We do want to absolutely recover the actual virus isolates and sequence those and begin to see what is the range of different variations that this RNA virus is capable of, just to keep track of whether there might be any variants that are going to associate with a difference in its severity. That data is starting to appear, but it's pretty focused in a few places. It would be great to have that kind of distribution of sequencing capacity to look at the genomics of this virus and how it varies over time and place.
Thank you. Looks like we have one other question? Terry, are you raising your hand again, or?
I am, but only to comment on the chat and perhaps ask Caterina Ferrecchio to comment. Caterina put a note that she has 10,000 adults aged 40 to 74. It's not quite clear where they are, but I'm going to guess Italy, who are in active surveillance. Caterina, could you comment a bit about those adults and how they might participate?
Okay. Can you hear me?
Yes.
Yes.
Okay. Hello. Yes. Well, this is an adult population. It's the population of small agricultural town. Was a population-based sample, and representing it was like 75% of the adults are enrolled, and we have a very close contact with them. And we just passed a small survey following an international recommendation about perception and symptoms. This small town only had 6 cases altogether since the epidemic began in Chile. So it's a very low risk population. But we are just beginning the epidemic. This is 200 kilometers south of Santiago. So this will be a place like for a vaccine trial that they are very collaborative. We have a very good relationship with the population. We are doing a nested study funded by CDC of bacterial antibiotic resistance in the families and how it is spread. We have a very nice infrastructure and biobanking. So it will be ideal for, I would say, vaccine trials, because therapeutic is not enough sample size. So it's open for.
Great. Thank you.
Okay.
So thank you, Francis. It was really wonderful to have you help us kick off this virtual summit. We know how extremely busy you are and appreciate any time we can get with you and to hear your insights as to what the IHCC could do. We're certain we will report back to you the output of the summit in many different dimensions. So carry on your great work. And again, thank you for all your wonderful support of our efforts here.
And thank you, all of you, for taking part in the IHCC, and especially to the leadership for having put together a really wonderful agenda. And I'm counting on great things to happen.
Thank you.
Great.
All right.
Thanks.
Bye-bye.